Thank you everyone for joining us today. Hopefully everyone, although we have very different weather across the country, everyone is safe and warm. It is my great pleasure to get to introduce Dr. Marcus Kaul, who is a professor of the UC Riverside School of Medicine, Division of Biomedical Sciences. Prior to joining UCR, Dr. Kaul was an associate professor in the Infectious and Inflammatory Disease Center of the Sanford Burnham Presby Medical Discovery Institute in La Jolla, California, and held an adjunct appointment in the Department of Psychiatry in UC San Diego. He completed his education and residency in pharmacy and pharmacology of industrial quality control and obtained his Ph.D. degree before coming to the United States in 1997 for his postdoctoral fellowship at Children's Brigham Women's Hospital and also with Harvard Medical School in Boston. In 1999, he was recruited to the Burnham Institute in La Jolla, California, and has since been working in Southern California. Is a member of many societies, many professional associations, and has a very strong interest in understanding the neuroimmune effects and neuroinflammatory injury in the brain with HIV infection. So please join me in welcoming Dr. Kaul here today to give his talk.
Thank you, Vega, for the nice introduction, and I'm happy to be back here. I'm just trying to my screen just changed. Let me just see. Let me just stop sharing because I need to. Let me see. Somehow my computer just. Okay, I think we're back. Okay, yeah, so today I want to talk a little bit about some work that we've been doing for some time now that relates really to a neuroinflammatory brain injury in HIV infection, even with cART available. And oops, why? Sorry, I'm having some technical difficulty here. Oh, okay, so it would be so. But before I get really into the science, I also want to mention show the people who have over the years really worked with us. And this is not only in the people that are in the group right now in Riverside. We started a lot of this work while we were still at the Burnham. We're collaborating a lot with our colleagues in San Diego over the years, and we have had numerous collaborators also outside, for example, at the Scripps Research Institute. And of course, for our work, very important, you will see is also the animal facilities in in all the institutions that we have been working at. And another really important part for the work that I'm going to talk about is actually the NNTC. And in particular, I have to thank Ben German with helping us there. And of course, nothing would working without NIH funding. And in the current situation, we hope that this will hopefully be resolved and continue. But yeah, so over the years already, we have really a lot of help there, and this is really, really highly appreciated. So just to get back, for those who are not familiar, many of you have seen this already. Some of these data, they come actually from a lot of this is from a review that was and my colleagues at Johns Hopkins. But the data, that are still out there. So the prevalence of HIV infection is still pretty high in the US, and these numbers have been, just pretty similar for many years now. We still have 1.2 million people in the US that get infected or are infected. There is more and more viral suppression in there, but we also, not everyone is suppressed. Not everyone gets to treatment, but the numbers are getting better. However, even if people are well treated and and even suppressed people, they still may experience some of the neurological complications. Early on in the pandemic, in the epidemic, really, people were referring to this as neuro-AIDS. We're now talking neuro-HIV because with in the era of cART, dementia and neuro-AIDS is a little bit less often, but what we still see is neurocognitive impairment, and we often now talk more about neurocognitive impairment in general. However, there was like 17 years ago, there was a new classification established as HAND, HIV-associated neurocognitive disorders, with these three clinical categories, the asymptomatic, the mild cognitive, and really the full-blown dementia in there. Now, here are some of the numbers, and this is really taken from this review article from Saylor, in 2016, that pre-cART, there were still almost half of the people had no impairment, but we had a significant segment of patients who would develop dementia and had minor cognitive impairment, and also some asymptomatic one. And with this arrival of very potent antiretrovirals, the dementia, the part of people who develop dementia is luckily much slower, but we still have a significant amount of minor cognitive impairment and an increasing amount of asymptomatic, so very mild impairment. And so the people who actually do not show impairment is still about only half of them, right? So we wonder why, even within suppression, with good viral suppression, people still develop neurological complications or neurocognitive impairment. And this is a major driver for us still to better understand what the mechanisms are. It's also a lot fewer people, of course, now come to autopsy, but even at autopsy, there's still, if people come to autopsy, there's still people that show, some of the classical signs, loss of dendrite synapses and gliosis. So we in the lab have been over the years, so we really like to collaborate with the clinicians and listen to what the clinicians tell us and do this. But in the lab, we're really basic science bench scientists and work with in vitro models and tissue culture models and mouse models, trying to figure out what is happening at the molecular and cellular level. And here is again a slide that many of you have already seen, but let me just to provide a kind of major framework that is still true after many years, and that summarizes the work of a lot of people. Many groups have contributed to what we know now about this. So HIV infection always starts in the periphery, right? And there's been a lot of work, a lot of people have worked on how does the virus actually makes its way into the brain? And the HIV infection that occurs in the brain and seems to go relatively quickly, can be within a few hours, apparently, after initial infection, might be driven by either infected macrophages or CD4 T cells that carry that, extravasate and, basically infiltrate the brain. However, they do seem to be, able to basically transfer the virus and infect some of the microglia, the resident microglia and macrophages, as well. And even if these are not a lot of cells, these cells, are basically productively infected cells. And pretty soon, if we, go by what we see, in the models or in vitro trials, these cells pretty quickly become neurotoxic. They release factors, besides, if they're infected, they release viral proteins. Some of those can directly act on neurons, but they also release other factors that all seem to be toxic to neurons. And what's been over, again, the work of many people, shown that the neurons eventually get into trouble with their neurotransmission transmission activity and also calcium homeostasis. So as a result of this, they lose some of their synaptic connections, or they shut down some of those in there. However, the infected cells do not only bother, so to speak, the neurons, they also, of course, interact with the other cells, in particular the astrocytes. Even some of the viruses seem to make their way into the astrocytes. We don't not yet completely understand all this, but the data are clear that you provide DNA in astrocytes in there. But the astrocytes can also, even if they are not carrying any virus, are affected by factors released by infected cells, and these are cytokines, chemokines, possibly, and they affect the astrocytic function, which changes then the way astrocytes, handle or help with neurotransmitter function. They may even lead to an increase of glutamate release, or they may reduce the production of growth factors. They also may actually produce some oxygen radical or like oxidizing radicals that also, compromise neuronal function. Now, an interesting part of this, and this is why, we appear to need only very few cells actually to cause trouble, is that microglia don't need to be infected in order to become neurotoxic. They just need to see apparently some of the viral proteins, and the same seems to be actually true for macrophages. So that is something that may explain why you have kind of, with very few infected cells, a kind of an amplification effect in terms of the damaging mechanisms in there. Okay, so this again is probably still a simplified scheme of here what we think is saying, but I think it sets a nice framework. Now we've been trying to figure out, so what is happening in the cells, and we have been particularly interested, of course, in what the macrophages and microglia do, because they are considered to be the major perpetrators here in terms of the toxicity. But we also, of course, are interested to see how's the reaction of the astrocytes and neurons in particular. So is there anything that can be done perhaps to protect the neurons? Now, one way, of course, in practical terms that it's well known is cART. cART is really important to prevent infection and propagation of the virus, right? But at the same time, the issue with cART is none of the drugs actually prevent an already infected cell from producing viral proteins. And we had this discussion in some of the other meetings, or any other speakers have mentioned that as well, that this, creates a potential or may, provide an explanation why even with cART, or even suppress viral production and propagation, that if you have a reservoir of cells that are already infected and you cannot prevent these cells, we have no tools at this point. We need to prevent these cells from still producing some viral proteins. And again, we know that some of the cells that macrophages migrate just need to see some viral protein in order to become activated and become neurotoxic. Now, while trying to understand really what is happening at the cellular level, we looked at what many people are looking at, cellular signaling, right? And here, this is basically, taken from cell signaling, courtesy of cell signaling, really. So there are these major pathways that are, prime candidates first to look at what is happening. And we looked over time at many of those. These are just the myogen activate protein kinases here. They're important for many, many aspects, anything from growth and development, but also very important inflammation, apoptosis, differentiation in there. And this is why we're really interested. And we are particularly interested here. I will talk about this in this candidate here. And this is the P38 MAPK kinase, where there's 4 isoform alpha, beta, gamma, delta. And we are particularly interested in alpha, because that is the kinase that has been linked to inflammation and apoptosis in the past. So just to go back, why would we be interested in that, right? And let's go back a little bit to some of the work we initially started. So what we see here on the left side is rat cerebrocortical cell cultures that contain neurons, astrocytes, and microglia. What is stained here is microglia and neuron. And what we see this is a staining for phosphorylated P38. And we see there is all the baseline levels in neurons. There's some baseline levels in the microglia that we can see here. Can you see my cursor? Okay. So can see there is some of this is here at baseline. However, if we incubate these cultures with GP120 alone, very quickly, so we can see here that we get a strong increase in phosphorylated in the neurons, but also in the microglial cells. So this is very nicely reproducible, and it gave us a reason to think, so what's happening? So we looked a little bit more into this. And we have here other staining stories, and then we used a kinase assay here first to see, for example, what the activity is. And if you can see, there is in the beginning of the time course a pretty early peak, then actually the activity goes down, but by 3 hours we are back up. And then basically at the end of the experiment, here's a 24-hour experiment where we look at this, the activity is up again. Now this is interesting because if we quantify how many cells actually have, at the end of 24 hours in the controls, the numbers don't change, but actually with GP120, we see toxicity, so we lose some of the cells, right? So at 24 hours, we end up basically having, we're having fewer cells, but we have higher P38 activity, right? So that means the cells that survive the toxicity actually end up having higher P38 activity. And this is interesting. So the cells apparently, so we'll see. And when we actually look where a lot of the P38 is, so we can see here stainings for P38 per se, and then it's interesting. So this is the mixed cultures where we see it's not that we have neurons with synaptophytes in here to show here. If we deplete the neurons, for example, with NMDA, and then look, actually the remaining glial cells have a lot more P38 relatively in terms of protein, but very little of the activated P38, the phosphorylated P38. If we deplete just the microglia, it doesn't really make too much of a difference because they make up about sometimes less than 1% of the cells in the culture. So there's clearly P38 is present in all the cell types, but most of the activated the active P38 actually seems to be present in the neurons and apparently also in the microglia, even if quantitatively these cells make up a smaller number. So wondering, so what does this really mean? So we set up more experiments, and in these red cerebrocortical cultures, we usually, we sometimes deplete the microglia there. And we can do this with ester. This is a very old tool to do this, and then we basically prepare, expose them to the GB120, then prepare cell lysates and do the kinase assays. So when we do this, we can already see here, this is the control when we have neurons and microglial cells there. But if we deplete the microglia, we can see we have in the total culture a lot less of active P38. So even though the microglia make up only a small portion of the cells, they have a significant impact on the overall activity that we see in the culture, because most of what the active P38 that we see here is actually derived from the neurons, right? So that means this is also linked basically to the toxicity, because at the same time, if we deplete microglia, we no longer have neurotoxicity in these cultures. So in order then to go more into what exactly P38 in microglia and macrophages does, we set out to knock down P38 alpha with siRNAs in these cells. Here in this case, we used THP1 cells and as a macrophage model, macrophage microglia model. And then after we knocked down P38, we incubated them with GB120 to create supernatants, and then the cell-free supernatants were transferred onto neurons and astrocytes, and we assessed then neuronal survival. So this is a busy slide. Let's be regular. This is just showing that the knockdown of the P38 worked. What we can see is P38 is not completely knocked down. It's like between 50 to 70%. These are 3 different siRNAs that work with this. But when we then look at the neurotoxicity experiments, so we see here, this is controlled survival. This is survival with the GB120 supernatants in there. And we see we lose a significant amount of cells. But if we knock down P38 in the macrophages, we no longer have this toxicity. So the NMDA here is the control for maximum number of neurons that we can kill in these assays. So that basically showed if we knock down P38 in the macrophages microglia, we no longer have this toxicity. And the interesting part, of course, of this is that we don't seem to be, it doesn't require apparently P38 to be completely gone. The knockdown of 50 to 70% is basically enough. So not all the P38 in the macrophages and microglia might actually be contributing to toxicity, possibly, right? But it's also, if you think about therapeutic approaches, is you may not need to necessarily, completely get rid or delete P38 in order to have some protective effect here. Now, of course, I showed you before that P38 is also present in the neurons, and the question was, what role does it play really in the neurons? So in this case, what we did, we incubated the neurons and astrocytes that would receive cell-free neurotoxic media with an inhibitor, pharmacological inhibitor of P38 or vehicle, and then exposed them to the neurotoxic supernatants and looked at neuronal survival. And what happens then is shown here. So we see here the supernatants, the controlled supernatants. And again, actually, if you with and without the pharmacological inhibitor for P38, it doesn't really make a difference. And again, if we have this supernatants, the toxic supernatants without inhibition, we lose neurons. This was done here with 2 different GB120s. But if we block P38 in the neuronal cultures, we also no longer have a loss of neurons. So this was very important one indication that P38 is not only important for producing the neurotoxins and the macrophages microglia, but it's actually also really important in the response of the neurons to the neurotoxins. And it's important basically for the cell death on the destruction of the neurons apparently. So it has a dual role really in this context. So again, summarizing a lot of this, what we basically see, macrophages and microglia if exposed to HIV, GB120, the same might actually apply to other viral proteins, and we know now also even to infection. Even if it's CCR5 or CXCR4 preferring GB120s, we get involvement of P38. We know that there is some of the steps we do not understand all the steps yet, but we know, and work by others has also implicated other proteins in there, but P38 is really the core here. And we get activation of these cells and producing a neurotoxicity. If the neurotoxins are then released onto the neurons, we understand there is a link that the neurotoxins, affect neurotransmission and NMDA receptor calcium homeostasis. Again, we do not have all the steps how the neurotoxins actually affect this, but even in neurons, P38 remains a critical gatekeeper that decides whether or not the cells will go into apoptosis and die. So, having that said, as we were wondering, okay, how does this happen? What's happening? Because P38, does a lot more. So we went back because actually it's interesting if you look how P38 was initially first discovered. It was discovered as by testing drugs that would prevent the inflammatory response of macrophages to LPS, for example. So we went back actually and did some of these experiments with LPS and with GB120. And this really summarized here is a larger microarray experiment at the time. We have exposed these cells to SPS with and without the inhibitor for P38 and with GB120 with and without the inhibitor. For 4 and 24 hours, I show here the 24-hour data. We then collect the simulations and isolate the RNA of cells and ran this microarray. And make a long story short, what we see here, there is GB120, causes some changes in genes, but all the changes were really, really moderate. Nothing compared to what, for example, LPS does in this numbers. But there were still a lot of genes that were changed. So if we look at some of those here, there are actually several candidates that are affected and that are linked to inflammation from IL-1, CCR4, 5, CCL3, CCL2, IL-1 beta, TNF, and so, but interestingly enough, these are very mildly upregulated in the context of GB120 exposure, but they, interestingly enough, even more upregulated in the block P38. That was a little bit a surprise because we thought, wasn't it supposed to downregulate if you block P38 to downregulate all this thing? So we do not yet have all the answers, and we're just trying to figure out what exactly this means. And of course, we're looking here at RNA levels. So there's still a lot of work to do in following up at the protein level. But these are actually genes that we checked out because they were mentioned in the context of potentially HIV infection, but they're not the most upregulated genes. If we look at the most upregulated genes here, either with LPS or with with LPS or with GB120, you see, these genes are more like in the middle range. And initially, when we first did this experiment, this is a while ago, the student who did this experiment, the Katie Matters, was really surprised, was kind of disappointed that GB120 wasn't really doing more on this. And so we were looking at this, and we looked at what else has been mentioned in the context of HIV. And then we found that one of the most downregulated factors here in these cells due to the blocking of P38, which we know is neuroprotective, right, and prevents, was actually a component that was ATC4S, right? And we were wondering, so what is that? So, and if we, go further and look over here, we have this Alzheimer's is really the major, and this was actually mentioned by Andrew Albright and Francisco Gonzalez Garano in one of their papers, where they looked also at HIV-related, differential gene expression in glial cells. And they had actually found that this protein was more upregulated in those cells. And we'll see that this occurs too. But we were wondering, okay, so if not so many of these factors are upregulated, significantly by GB120, but there are some of these factors that are downregulated. So maybe this is how the protection of the P38 inhibition could work, not by preventing necessarily upregulation or even, upregulating some effects, but actually by downregulating a certain factor that's constitutively expressed there. So this, question then really led us onto a longer project. And just to summarize of this, is really led us to look at to this pathway. And this is here about really how we get from HIV to cystine leukotrienes. And then you may wonder, cystine leukotrienes, what? When we first found this, there was really nothing known about cystine leukotrienes in the brain, and very little in the context of HIV. So in fact, there was a paper in 1992 where with Howard Ganelman's group and David Wolski, where they had looked at macrophages in HIV and inflammation, and they noticed that HIV-infected macrophages were actually producing one of these cystine leukotrienes. They looked in a very elegant method at this time, actually with radioactive biosynthetic labeling. They showed that there was increased production of LTD4. So just quickly, so what we know, what this pathway now, and again, this summarizes some of the work. A lot of this is published now, that we basically go from HIV or GP120 in both cases, through CD4 and the chemokine receptors, we have P38 activation in there, right? And then we get actually to cytoplasmic phospholipase A2, and this is the arachidonic acid cascade really that gets involved here. And we get then from the lipids through several factors 5-LO and FAP, and then we get here to LTC4S, right? And LTC4S is the key enzyme to producing cystine leukotrienes. Cystine leukotrienes are then released into the supername, and in the supername and outside extracellular space, they are quickly processed by peptidases from LTC4 to LTD4 and LTE4. Now, these then can bind to several receptors. And initially, for the best known is really cystine leukotriene receptor 1 and 2. These receptors were only later actually involved, or more recently linked to this. But again, in the context of HIV or the brain, there was very little known. However, cystine leukotrienes are known to play a really important role in asthma. And this is actually something that were in a way lucky, because there was already a drug when we discovered that there was a drug that's very well established in asthma therapy, montelukast, that specifically blocks cystine leukotriene receptor 1. And to make a long story short, and you can look some of this, we found then that so if you use montelukast instead of for example, the P38 inhibitor in one of these neurotoxicity experiments, where we transfer cell-free supernatant from neurotoxic supernatant from HIV-infected macrophages onto neuronal glial cultures, we get the toxicity with the HIV supernatants. But if we incubate neurons and astrocytes with montelukast before, we no longer have significant toxicity. So we really have a protective effect. And that basically indicated that this pathway seems to be a critical component of HIV neurotoxicity, right? So it leads us back to, so there is definitely an inflammatory component that we know from asthma that apparently can also play a critical role in the brain in the context of HIV. So, and you may say, okay, fine, but this is all in vitro and model experiments. So is there any indication that this plays a role in real life? So this is actually when the NNTC became really important to us here. And again, Ben Gelman, who was had been looking at us, and we had some discussions. So he provided us with a set with samples of a cohort of patients. So these were a total of 134 samples. 60 of two of those are non-infected controls, and 72 are from patients. So HIV patients. And here it is, these are earlier, some of those were earlier in the HIV epidemic collected, some later. So these patients, the majority of these patients has been treated with cART or earlier versions on this, but very few of these patients are actually suppressed, or so most of these patients actually, at the time the samples were collected, did have detectable viral titers. So, okay, so this, what the NNC provided us, Ben Gelman's lab actually prepared RNAs from these, and so we could actually look at this. And this is the same cohort that actually other people have studied as well. I mean, Ben Gelman published on this cohort Two, Dennis Colson's lab did a lot of work on these and a couple of others. So we collaborate with Adam Fields on some of this analyzing in the context of some other factors. But coming back now to our question of the neuroinflammatory part, and in particular, LTC4S and cystin leukotriene receptor. So when we analyzed, and this is really the RNA, here at the RNA level expression, you see, it may not be super like specific, or very high correlations, but we found significant correlations between LTC4S and CD68 here, when we consider as a marker for the myeloid cells and macrophages on microglia in there. So on this, so they correlate. But we also found actually surprising that cystin leukotriene receptor expression correlated with HIV DNA, viral DNA, as well as the RNA in the brain. So, and that was another indication that this inflammatory pathway may have a role here in what is happening in the HIV-infected brain, and also how it relates to injury. In fact, if you go back and look at the paper that is published, you can see that brains that show signs of neuropathology have significantly higher expression levels of cystin leukotriene receptor. Okay, but we expanded and looked at other components here that are related to this, and there is, for example, also a pretty good correlation, significant correlation between DUS1 and LTC4S. DUS1, so what is this? It's a dual-specific phosphatase that is actually very important for regulating the activity of P38, right? And P38, on the other hand, regulates LTC4S expression. Also, I didn't show you the data here, but it's in the paper. So if you block P38 and at the RNA level, you could see that LTC4S was downregulated, and this holds true at the protein level as well. So it's a regulator. So that might be, perhaps this is a negative feedback. We can't say this for sure, but there is definitely a correlation in the expression there. Of course, correlation is not causation. What is perhaps less surprising is that there is also a correlation between the MAP kinase 14 and DUS 11. So MAP kinase 14 is actually P38 alpha in this case. So that makes sense because they are basically a regulatory gene expression or regulatory pathway, and also in terms of activity, because DUS dephosphorylates P38. And that's basically downregulates the activity of the kinase. It doesn't regulate the expression of the kinase, but it does downregulate or affect the activity of the kinase. Now, one thing then that was really surprising and very interesting to us was when we looked in these data, because not all the data in these NLTC samples have full neuropsychological assessment, but a subset of this has. And when we looked there, we found a really interesting and surprising finding that actually the expression level of P38, again, this is just the expression level, it's not the activity, but at least expression level, correlated with the global deficit score. So that was another reason for us to see, okay, so this kinase really deserves a little bit more of our attention and may actually really play an important role here in the context. So we set out to look more at this and went actually back to our models. And some of you know these models because a model that we have been working with for a long time now. It was initially generated by Leonard Mookie at Scripps, and we have spent quite some time characterizing this model, or a little bit more, simply to see what this model can actually do and what it maybe not be able to do. But what we see there is the loss of dendrites, synapses. We see activated microglia, astrocytosis at different ages. We also know that there's compromised neurogenesis. There has also behavioral changes. There are electrophysiological alterations. There is also endocrine system alterations. And most interestingly, and that's what we find in the larger scale microarray study a few years back, that there's a significant overlap in differentially regulated genes with the human brains. Basically, so we compared this to the NNTC brain array, and this is where we found that there was a lot of overlap. So these are all basically key features of HIV-induced brain injury also in humans. So, and this was one of the reasons why, or major reasons why we actually decided, okay, let's use this mouse model to dig a little bit deeper into what the kinase might be doing in there. And just to look at some other data here. So this is the data that I was just mentioning before. If we look at the NNTC cohort and compare the non-infected versus the infected one with brain pathology and the ones without brain pathology, we see that the ones with brain pathology have these increased levels of cysteinyl leukotriene receptor type 1. If we look in the mouse model over time, and this is here in the microarray, we can see there is over time also an increase in the cysteinyl leukotriene receptor. There is also, the flap is the protein that activates actually the arachidonic acid cascade and the cysteinyl leukotriene synthase here. That protein is also follows the different is higher expressed in, compared to the control, but we can also see that the cysteinyl leukotriene receptor. So we have here again a similarity with what we see in the human brains with brain pathology as well as in the mouse brains that also show pathology. So in also just we looked at the brain and the activity actually, we just wonder, okay, so it's all the expression, but is there activity? And this is here we used a kinase assay for this. So this is an immune complex kinase assay. I mentioned the kinase assay before, basically where you lyse here brain cells or brain tissues, prepare an extract, you immobilize, then you precipitate this in an antibody, and then so you have then the active kinase bound to a beads as an antibody, and then you expose those beads with the active kinase to an indicator substrate, which is in this case a recombinant ATF2, and then this ATF2 you can, the phosphorylated ATF2 you can detect with immunoblotting. So it's a non-radioactive phosphorylation kinase assay. And you can see here that in GP120 expressing brains, the activity is significantly higher. So we also have higher P38 activity. We have meanwhile confirmed this also with Western blots of phosphorylated P38 in there. So, and this is also that is published in there. So we have, so clearly, P38 is present there and the activity is also higher in the brains, right? So, how would we go about doing this? So remember, the microglia are really considered the main perpetrators in terms of neurotoxicity. So we said, okay, let's try to target those cells in the brain. And so luckily enough, there was a fluxed MAP kinase 14 mouse available. It took us quite some time to get it. Was even so it was available, so we didn't make it ourselves. But even getting the mouse, it was initially made in Japan by a group in Japan. The investigator then moved to Europe, and then so eventually we found out that the mouse was actually already in San Diego in a different lab, and so it took a while, and eventually we got the mouse, and then we crossed them into the GB120 mice, and in order to get the microglia-specific knockout, we used here the CX3CR1 CRE mouse that is very good. So it's basically direct the knockout basically to the microglial cells. So we ended up getting here like 4 genotypes. The expressing mice, the wild type that do not have GB120, then the expressing cells that don't have CRE but GB120, and then the MAPK9 just CRE expressing mouse, and then the MAPK 14 cells that do express GB120 and the CRE. So, and these basically then result in a P38 knockout in the microglial cells. So we know this, these are our controls, the wild types and the neuropathology, but the question was what happens in those mice, right? So we first looked here, this is isolated microglia from those brains, and then, with the FACS analysis, just to show what we have here. And then in the Western blot, you can see of those microglial cells that P38 is really knocked out, it's really gone. This is GAP-DH as a control. So we then also looked, how many of the microglia do we have? And it's interesting that despite the knockout of P38 in the brain, the number of microglia actually didn't really change too much. We have, there's not too much really happening in CD11. B is still upregulated in the cells, even with the knockout. IBA is also still up, and so the P38 knockout didn't do anything around. Interesting enough, for sure, so is that if we look at the knockouts, that it didn't change anything in terms of GB120 expression. And it's perhaps not so surprising because the GB120 in this mouse is actually expressed in the astrocytes as a soluble protein. So again, it was perhaps a little bit surprising that the P38 knockout, did not change some of these markers, and in terms of that also are linked to actually microglial activation. Okay, so when we then put the mice actually through behavioral testing, and it's just showing some of the Barnes maze here, you could see that actually the mice in which the GB120 expressing mice that lack P38 in the microglia made less mistakes, and they actually also did better in the Barnes maze in terms of what we see here. These are the GB120 expresses mice that do have memory issues. The spatial memory is reduced. They spend significant more time in the area in the quadrant where the escape chamber was. But actually, if you knock out P38 in the microglia, the mice are almost as good as, not as good perhaps as the wild-type control, but they are doing pretty well in there. So basically, that indicated already, okay, that knocking out P38 in microglia might actually help brain function and memory function in this case. Now then we went back, and this is a longer histological analysis. And to make a longer story short, this is what we see usually in the GB120 mice in terms of losing MAP2 positive neuropil, meaning it's an indicated injury to neurites. But if we knock out P38 in the microglia, this loss no longer occurs. And the same is true for synaptophysin. If you look at synaptophysin in the GB120 expressing mice, it always have a significant loss. But if we knock out P38 in the microglia, that loss no longer. So that was then really first time in vivo evidence that P38 in microglia, and just in the microglia, in vivo, seems to play an important role in this kind of injury that we can see. So the same, we looked, and then also in terms of more neuron or loss of the neuron. This is looking with neuron positive cells in the cortex, as well as in the hippocampus. And in a similar picture, you see some loss with GB120 that no longer occurs if you knock out P38 in microglia. And the same is basically true in the hippocampus. So all this really indicate that if you take this together with the behavioral data, it's looks like, yes, there is clearly some protection. So MAP kinase in microglia seems to play an important role. It's interesting, then when you look at the microglia themselves, because we still see with GB120 we get an increased number of microglia, and that actually doesn't change. So even with the knockout of P38, even if we don't have neurotoxicity, we still have more microglia. And this is often, in terms of pathology, considered to be an indicator of activation, or that there is trouble in the brain in there. Now, the same is true if you look in the hippocampus. So the numbers of microglia and this level of activation, or the proliferation that occurs, doesn't seem to change there. Now we went back and looked at then what does it mean for the gene expression. So we isolated again microglia, and this is not single cell sequencing, that is actually still bulk sequencing at the time. And there's still a lot of follow-up work to do, but just to find out, what the differences are. If we look at this here, if we look at the GB120 versus the wild type increased expression in the presence of microglial P38, we see that, for example, just to mention here one of the factors, CXCL13 is upregulated significantly in there. And then actually the upregulation of CXCL13 has been linked to detrimental, course of HIV infection in there. However, if we knock out P38 or no longer have P38 in the presence of the GB120, then CXCL13 is actually significantly downregulated. So that fits with the notion, right, that the knockout of P38 in the microglia has a protective effect, and also perhaps beyond protect the neurons, also in the entire course of neuron. This is a little bit hand waving at this point, but I think it's just to show one example of what is what might be important in the future to follow up to see how these effects what these effects are. So to summarize and to finish up here, so where are we at this point? So we know now, we have some evidence that GBO1 and HIV infection per se, but also GBO1 can trigger some of this pathway here is really P38 in the microglial cells. And we know that the microglia release other viral proteins, other factors that are toxin and toxic, including some of the excitotoxins, and they also release cytokines, right? And these might directly work on neurons and various neuronal receptors. However, it seems that the arachidonic cascade downstream of here, and in particular the cis-leukotrienes, do play a critical role here. And again, these cis-leukotrienes can act on neurons as well as on astrocytes. And what their role is, what is more detrimental? Is it the direct interaction with the neurons, or is it, is there some change? This is something we still have to figure out. But we know that already that P38 in neurons is very important from wherever the injurious, mechanism is, and from where it comes. That P38 in the neurons plays a really critical role in the decision whether or not these, cells in response, have this synaptic and dendritic injury, and eventually go into apoptosis. And there. But then we know that astrocytes also express P38, and we know that there is a transient activation of this. How much that actually plays a role, we still have to find out, because we all know that astrocytes, of course, are really important in in handling of neurotransmitters. And we know from other studies that that might also play a critical role. So it's just not one factor here that decides whether or not the neurons, make it. So, or this is basically the injury that that HIV might be able to trigger. But we have, I think, now a little bit an idea what some of the critical components are that contribute to this injury. And I think I will end here, and we'll be happy to answer questions.
Thank you so much, Marcus. That was a fantastic talk. I'd like to open it up to questions if anyone wants to unmute. I do see there's one in the chat.
In the chat? Okay, let me see if I can, get the.
Asa, do you want to unmute and ask your question?
So, okay, so Scott, there are too many outliers on the first question. I thought of my friend. Yes.
So, just being nitpicky, it was on those graphs that were on the slide that was before the list of transgenic mice references. So,
yes,
a big cluster of points here, and 4 points, 3 here and 1 over here, and you drew a line through it.
Yes, so it's a good point, and this I think is a fair point, because we had a fair amount of discussion about this also to see, right? And then so also with some statistician, and in the end, so we used the route what GraphPad offers in terms of determining outliers, and these were the values that came out of this as being should be considered, right, in there. So but I'm open to discuss this, to see if I mean statistics doesn't end the data, but it's important for us to consider how we interpret some of these things in the. And of course, certainly we would now really like to look at more samples just to see if we can confirm that, right? In more specimen.
All right. So that is a very minor thing. Beautiful presentation, of course.
Oh, thank you. Well, so Fatah, you had the question. Have you looked at complex size changes after? Oh, so that's a good question, actually. So yes, so this is a whole story, and it's all where is the active P38, right? I mean, if you look at the immunofluorescence staining, you get this kind of granular staining, right? And so depending on how good your antibody is and how much you have, you can see, yes, there seems to be certain locations where the activity seems to aggregate. So we've actually been trying to get a proteomics approach onto this, and it's what we find that the complex in the cells that you can precipitate actually with an antibody for the kinase assay is amazingly stable. And so in this in this complex, the kinase activity actually persists for actually for days. You can keep this, and you can come back days later, and it's still. So we were, of course, you know, you in the assay you put in the phosphatase inhibitors, right? But you wash away. So, but no, to get to the answer, so we do have some indication that there is a number of proteins that are associated with active P38, but it needs more work to figure it out. We do not know exactly how big the complex is. There we haven't done any size exclusion. That is a nice idea to do that. But we were surprised because how stable this, you know, when this complex really is.
Very nice.
But it's a good question, and it's a good thing, yeah. We are actually, so we're looking, we're trying to look at some other ways, and I hope I will have, you know, something, because the question is, the big question with P38 is, of course, how does the kinase that sits, you know, is basically present in every cell, distinguish between the signals that lead to neurotoxicity and other signals, right?
Exactly. Yeah. Thank you.
So there is, let me see, is there,
Amanda,
yeah, so
has a question.
Amanda and Mary, I don't know who was first.
Amanda was
I'll be quick. Thank you so much for outstanding talk. I'm just always so jealous of the wonderful animal models, the things that you can do. I was curious about the montelukast.
Yeah.
Is that FDA approved? And just
Yes,
thinking
it's
about
FDA approved
people.
for asthma treatment.
Oh,
For asthma
okay,
treatment.
interesting, right? Because when we think about these legacy effects, you know, in terms of, you know, the viral proteins, right? You're focused on GP120, but, you know, other proteins that might be expressed in the brain and ongoing damage. So do you think that, you know, this could ever be, you know, go to a clinical trial or to see if it could further protect, especially when we're aging,
I mean,
you know, all the issues. Thank you.
Yeah, you know, I mean, so to be honest, I was wondering, and I'm still trying to figure out, you know, how many, you know, it's so widely used for asthma treatment, right? That you would wonder if there aren't, you know, HIV patients who may have asthma that are using it, right? And so far, we haven't really gotten to get to them, because usually the patients, you know, evaluated for HIV are not necessarily apparently, you know, in the same way evaluated for asthma. And in asthma, you know, people who care about asthma don't seem to be involved in any way of HIV care, right? So it would be a question I would have for our clinical colleagues, you know, if they know, or anyway, I think it would be really interesting to see. So far, we cannot really tell. So we're working actually on, you know, we did some, because the nice thing about montelukast, it's actually orally available. You can take it orally, and we have so we've come up with a way actually to feed it to the mice, and we are now actually so we did this, and we're currently analyzing, you know, to see if that would work. But yeah, I mean, it would be I mean, of course, you know, think maybe it would be worth. I don't know if anyone has montelukast tested ever in the SIV model, but maybe that could be a next step. So I don't know enough about monkey asthma, and I don't know if asthma in monkeys is actually you know, a clinical or veterinary you know, problem that, you know, that is measurable. But it's a good question, yeah.
Mary Jane, would you like to unmute?
Mary Jane, yeah.
Yes, I apologize. I'm on a foreign laptop and I can't get the camera on. So like everyone, I really enjoyed the last hour. But I have a question. It was in the last data slide of the RNA-seq. And there appeared to be quite a number of interferon-related genes expressed, except I missed out, was that overexpression in the GP120 transgenic mice.
So, sorry, which one did you miss?
It was the last data slide, the RNA-seq. There was induction of a number of interferon-related genes, but I'm unclear who is the numerator and who is the denominator there.
So you mean who's where this is coming from? Yeah.
Where do you see the overexpression? What,
So,
Miles?
okay, sorry, yeah. So no, it's true. So you see in the GP120 mice, you do see an induction of interferon-related proteins in the microglia. So when we first looked at this in the micro area, it was like whole brain, and we could clearly see that there's an interferon response. But we largely thought, okay, because the GP120 is expressed in the astrocytes, so we thought this might be, and it is, still the original viral sequence that is expressed, and this GP120 sequence has lots of things that are known to induce an interferon response. We thought, that maybe it's primarily the astrocytes. But and we know that in the mouse, you have a transient expression, you see some interferon alpha, but you see interferon beta in particular in the beginning, and then it's transiently expressed, and then the expression actually goes back to baseline pretty quickly. It was in a few weeks. And so there is definitely, and I guess that what we see in the microglia might actually be driven by the interferon that is released, a soluble interferon, because the GP120 is not expressed in the microglia itself. And GP120 alone, for example, doesn't induce interferon proteins. The protein alone does not, at least in our hands, did not induce interferon related genes in microglia, so far as we can tell. So in the slide I was showing, so it was basically, so downregulation was with the knockout of the P38 in the microglial cells. And already, I mean, if you have so this was what the slide showed, was the comparison on the right side, showed was the comparison between the GP120 expressors and the microglial knockout.
Okay.
So what this also tells you, that it doesn't, knocking out the P38 in the microglia does not change all the different gene expression that you get with the GP120 expression, right? So that is and to protect neurons, maybe that is not necessary, right? Maybe there's just some critical factors that need to be differentially regulated in order to prevent the neurons from deteriorating.
It's terrific. Thank you very much. Thank
Oh,
you.
sure. Thank you.
There's one more question in the chat from Katie, if you would look at that, and then cognizant of everyone's time. Okay, like ask the question.
Sure, and I do realize we're running over time, and this is mostly out of curiosity, but I was just wondering if you've ever done any staining or looking at HSV co-infection in the NNTC samples or the human samples?
Thank you. That's a good question. No, we haven't. But I think that is that is a good question. So we had actually questions about, you were wondering about CMV, but HSV. Yeah, you're right. I mean I think
just with all the resurgence of the focus on HSV and the Alzheimer's type literature, and
yes,
there is a good link with the P38 and MAPK. There. So
you're absolutely right, Kenny. That's just a very good question. I totally agree. I mean, it would, we do have, we start wondering, also looking at COVID, looking at HSV, CMV, what has come up, exactly as you say, also this link in potential link to Alzheimer's, right? We were also wondering, what other viruses might potentially contribute to that, right? I mean, because I mean, if you would think that people living with HIV probably have exposed to at higher risk often to contract other viral infections as well.
And I'm sure they feed into each other, right?
Right, and you're absolutely right. Does it change? Because we are also so what is we're trying also to understand why do neurons have this relatively high level of baseline P38 activity already, and also microglia, right? If we compare it to other cell types, these cells have, compared to the level of P38 or the amount of protein that P38 protein that they express, they have really high levels of active P38. Whereas, if we look, for example, at astrocytes, they have lots of P38, but only a fraction of this is really activated. And you would wonder, if any other chronic infection, could feed into this potentially and have an effect on the baseline level. I think it's a really good question. Thank you. Yeah.
Awesome. Thank you. Awesome talk.
Thank you.
Yes, thank you so much, Marcus. This has been a fantastic talk. Just a quick reminder, there will be no seminar next week, but we will resume again the following week. So thank you all for joining us.
Thank you. Yep. Bye.
Thank you. Bye.